Hi, my name is Eric Kavi, and I'm with the Department of Electrical and Systems Engineering here at WashU. I learn about learning. The data will tell. Where models come from, I hope I do well. See, it's quite the challenge to pare down the time, but it's even trickier to do it in rhyme. It all starts with the data, numbers and letters, and the question therein. Can we learn better? The data we possess is not what we want. We must mold it, shape it, show it a merry jaunt. It is for this aim that I propose a tool, the machine learning morphism that I made in grad school. A building block of math with inputs, outputs, and priors galore. A function to learn, but wait, there's more. A risk under the hood, attached at the hip. It learns from the data, so models don't trip. With this simple structure, I do some weird math. Even adding things together has no clear path. If you make an application, just chain them together, end over end, like birds with no feathers. Let me give you a story to show what I mean, of patients and hospitals and models in between. There was once a man who got sick, who went to Barnes-Jewish right quick. They checked him for gout, then kicked him right out, and he readmitted again in a tick. The doctors didn't know what to do. They took measures and scores with no clues. The models were stumped, the patients were grumped, and to Uncle Sam, all the money was due. There was once a boy from ESE who thought this problem was easy. My code was wrong, but my spirits were strong, and then it was 2 years later. I divvied up the patients by their quirks, and used many models to fit the patterns that lurk. I made a cool graph, did some weird math, and now I have to figure out how it works. There was once a man who got sick. He went to Barnes-Jewish right quick. The doctors went on the attack, but he still came back. Because their new fancy system sucks. Sorry, I have to keep this PG. So, my captive audience, we come to the end. I would like to reassure you that your brains will mend. At this point, you should know that I do machine learning. And truth be told, that's pretty much the show. Thanks. Vote for me.
Cardiovascular disease is the leading cause of the deaths today in the world. When talk about why someone had a heart attack, what was the first word that came into our mind? Maybe exercise, diet, age. But pause there and take a look at this young man who's a gym trainer, taught people how to lose weight while he himself had a pretty deadly heart attack at a relatively young age. Next to him is a 100-year-old granny who's still enjoying her cigarettes. Stories like that basically told us there must be something about genetics involved in this disease. However, after decades of efforts, the scientists haven't quite solved the DNA puzzle because there are too many missing pieces. And my job during my PhD research is to find them. And I focus on structural variants, which are large-size DNA sequence differences among individuals. They are pretty much under study in cardiovascular disease setting, but in other contexts like cancer, we know that they can cause big damages, so they are great candidates for my research. However, they are also pretty rare because they're pretty deleterious and under a very large nature selection. So to increase the chance of identifying them, we collected our samples from people in Finland. And they have been through a special event called genetic bottleneck. Basically, their ancestors started from a small population in European, and then they expanded into a large population in a short period of time, like what I show here in the slides. So the deleterious variants like the blue and the yellow dots here are likely to reserve in the population with slightly higher allele frequency. And with this special setting and the computational method we developed in lab, I was able to find a lot of missing pieces. For example, we find that about 3% of the population carried a deletion next to the gene encoding albumin. And that variant also affecting the albumin level in their blood dramatically. Affecting albumin is like affecting the delivery truck for molecules like water and hormone and fatty acid, and therefore Lower level of albumin usually associated with higher risk of cardiovascular disease. So piece by piece, we're filling up the puzzle like this. And study like this not only helped us to understand the disease better, but also help us to improve the prevention, diagnosis, and treatment of cardiovascular disease.
Don't like the boring background in a virtual meeting? No worries, artificial intelligence can replace it with an image. How about the Golden Gap Bridge? More and more AI systems are on a way to assist our daily life, but operating any AI system requires energy. Tesla Autopilot is running on a chip consuming 60 watts of energy, more than 4 times the energy used by our entire brain, which is much smarter. If you want to reduce energy consumption, consider learning from our brain. Both our brain and AI system take in environmental signal and compress it. The compression keeps the important information and discards the redundant one. While watching this, huge volume of a signal enters your eye and reaches the retina in the back. The retina then compresses the signal down to only 1% of its volume, but with little sacrifice on the resolution, the amount of information. Amazingly, when we binge watch Netflix, our eye never get harder. I studied the strategies that neurons in the retina use to extract meaningful information and use as little energy as possible. One of the strategies used by the retina can be explained with the following metaphor about social distancing. Imagine in a grocery store, employees are restocking shelves before customers rush in. Due to COVID-19, they also need to maintain social distancing. The workers spread out to their workspace in the store, from fresh produce to packaged goods. But not everyone's space is allocated equally, because restocking efficiently is also critical. It takes longer to restock a delicate produce, so the workers there are assigned to smaller spaces. In the packaged goods section, it's much easier to restock bulky boxes, so workers there have a larger space. Similarly, in my physics, different types of neurons are spread out to encode diverse information from the environment. In a region with dense information, each neuron covers less space. In a region with sparse information, each neuron covers more space. This encoding strategy maintains a balance between the density of information and the size of the coverage, and it results in 15% less total energy consumption. My research demonstrates a way to maximize encoded information and minimize the consumed energy. To reduce carbon footprint, AI systems need to be both smart and energy efficient. The solution? It has long been inside us. Thank you.
Detecting disease at its early stage is very critical. In case of COVID-19, early stage diagnosis will enable timely treatment and isolation. However, accurate early stage diagnosis requires the detection of tiny amount of target disease molecules in your body. It is challenging because the signal is usually weak when the concentration of the molecule is low. Therefore, large and dedicated readout instruments are employed, which is very expensive and time-consuming. To address this challenge, we have developed a technique that can largely amplify the signal of target disease molecules. Now, you can detect the signals by just using your cell phone or even detect the signal with your eye. How is that possible? So, in most biomedical tests, a fluorescent dye is employed as a signal reporter, which indicates the presence and concentration of target disease molecules in your body. Like other signal reporters, the conventional fluorescent dye, the signal is usually weak. Here, we have developed a nano-sized structure that is composed of a gold nanoparticle highly densely coated with a conventional dyes. The gold nanoparticle acts as the antenna, largely absorbing and scattering the light. This highly concentrated light is then funneled into the conventional dyes surrounding the gold nanoparticle. And owing to that, the overall brightness of this nanostructure turns out to be 6,000-fold brighter compared to a single conventional dye. And now, you turn a dim matchlight into a bright beacon on the sea, which you can recognize easily. With the help of this ultra-bright nanostructure, we are able to detect extremely low concentration of target disease molecules in just a tiny drop of blood, which is 1,000-fold more sensitive compared to the gold standard diagnostic tools. Now, you can read the test result by just taking a cell phone picture. What does that mean? It means that the patient can potentially perform the diagnostic test at their home instead of going to the hospital. Imagine how that will impact on people in rural areas and developing countries who have very limited access to biomedical service.
3 to 4 months from now, most of us used to think that aerosol is this spray from cans like deodorants or disinfecting sprays. But thanks to coronavirus and of course news channels, now people have started to think that aerosols could be viruses or cough droplets suspended in air, which are really, really tiny, like 100 to 1,000th of the size of my hair. Nevertheless, spray from a can and viruses are just two examples of this huge world of aerosols. Broadly, they can be divided into good and bad aerosols, and you'll be surprised to see that how both good and bad aerosols impact our everyday life. Good aerosols are particles that you want to synthesize. For example, the paint on your balls won't shine, your sunscreen won't work, and your car tires will quickly wear down if it was not for these tiny particles added to them. On the other hand, bad aerosols are particles that you want to get rid of, like cough droplets, exhaust from engines, etc. My PhD focuses on understanding the synthesis of good aerosols and to capture the bad aerosols. The slide shows how I synthesized good aerosols using a flame reactor. I investigated how molecules come together and form these first tiny nanoparticles. I found that in very early stages, these particles not only interact with each other, but also with charged ions and electrons in the flame. The know-how of the physics of particle-particle interaction and particle-ion interaction is very useful in controlling the final product, which in my case is used for efficient drug delivery and medical imaging. In the second part of my PhD work, I evaluated a plasma reactor to capture particles and therefore cleaning as well as disinfecting the air. Plasmas are fourth state of matter where the gas is ionized, so all electrons, charged ions, and neutral species are present. The slide shows a very shiny and bright appearance, which is due to this high concentration of ions. Now, to capture these particles, you need to charge them efficiently, but nanometer-sized particles are very difficult to charge. So we thought, if we could provide high concentration of charged ions and electrons through plasma, maybe we could charge these particles. So I designed a plasma reactor setup in our lab, and by adjusting plasma power and plasma geometry, we were able to charge even nanometer-sized particles. And once charged, it was easy to capture them. In future, we plan to test this study to disinfect and clean air from viruses like influenza and COVID-19. In a nutshell, my PhD work explores the wonderful world of aerosols by synthesizing the good and capturing the bad aerosols. Thank you.
The age of computers has changed education forever, making online education highly popular and accessible. In 2018, about 33% of college students in the States were taking at least one course online. Even in traditional classrooms, the usage of online readings and video lectures has increased drastically. Furthermore, because of the current coronavirus crisis, the move towards online education fastened across all ages worldwide. It is undeniable that these practices increase how much students must regulate their own learning, using their own intuitions on how much to study, a concept called self-regulated learning. While self-learning, students pace themselves through the material, having the option to spend as much or as little time studying. Thus, in this new era of online education, a crucial question is whether self-regulated learning is effective compared to not being able to self-regulate. Despite its importance, however, the majority of self-regulation studies lack a control condition in which students do not regulate their learning. Hence, in my dissertation, I examined the simple yet critical question: to regulate or not to regulate. In my 3 experiments, students studied words of varying difficulty and then took a test on the material. To address my question, some students paced their own study, spending as much or as little time as they wanted on the material, whereas other students' study time was predetermined and fixed, not allowing for self-regulation. Critically, the total study time of both groups were kept exactly the same, so the groups could only differ on how they use their study time. Good news is that in the first 2 experiments, students did benefit from self-regulation on both easy and difficult material. As you can see on my slide, they even closed the knowledge gap between easy and difficult material through self-regulation. In the last experiment, I introduced effective study strategies such as practice tests and self-monitoring questions to see whether the benefits of self-regulation persisted even when other effective learning methods were present. These methods boosted the performance of both groups and eliminated the benefits of self-regulation observed in prior experiments. To summarize, my dissertation results show that students actually learn better when they regulate their own study. This suggests that within online education, pre-recorded classes might have benefits over live online classes because they give more autonomy to students. The disadvantage of live classes, however, can be alleviated through effective study strategies. These findings impact how we shape online education in the current crisis and for future. Thank you.
Now, think about your favorite literary book. Ready? What is its style? Why do you like it? Do you think most readers would agree with you? What about those from foreign countries or even the ancient time? No need to rush to the answers. I just want you to get a taste of the central concern in my dissertation: the relationship among literary style, readers, and certain historical background. My project analyzes the detective fictions in early 20th century China, which were very popular at that time. These fictions have a modernist style, which concerns more about people's internal world than the external physical world. By expressing the anguish in modern man's mind, literary modernism criticizes modern life. Studies of modernism are mostly about those difficult literature for the elite readers, as if they assume that the ordinary readers don't understand modern pain or they only read superficial literature. I challenge this bias by explaining why and how these detective fictions were both modernist and popular among the readers. The first step is to know who the readers were. I looked for information about the publication of the fictions, their prices, different people's levels of literacy and income, etc., and eventually pinpoint the readership to the middle-class city residents. Then I examined the historical background of the readers. I chose several historical factors that stand out from the fictions. For example, many of them talk about the use of science in investigation. Therefore, I looked into when and how modern Western science came into China, why it was popular, and through what kind of media the ordinary readers knew about it. It is in these specific historical conditions that fiction's modernist style expresses an ambiguous attitude toward modern society in a way that appeals to the ordinary reader's standpoint. Then it comes to the final question. So what? What's the point in writing a dissertation about it? In my own discipline, I expect to call for re-examination of modernism. But outside the academia, my primary goal is to show that these detective fictions are really a pleasure to read, and I hope more readers today will become interested in them. On a deeper level, this is a particular attempt to answer a broad question: why do we like literature? It may sound abstract, but it can be a start point for us to understand ourselves and our time from a humanitarian point of view. Thank you.